Hello, good evening to all of you. Welcome to Unacademy YouTube Live. Hello, how are you? Are you fine? So here, today our topic is anatomy of flowering plants. Anatomy, internal structure of the plant pods like leaf, stem and root. And also about different kinds of tissues and the structure of each and every tissue. And also what is the internal structure of the root, stem and leaf, dicot stem, dicot root, monocot stem, okay, like that. And also you will be having this topic, secondary growth in plants, okay, and what type of questions will be asked from this topic, which are very important for EAMCET and also other type of competitive examinations. So your attendance is very, very important. You are given very good chance, okay, to watch the live classes so that you will be getting benefited. You will acquire, okay, a good knowledge regarding the different topics and the questions what are very critical, which need to be explained by educators. So this is very good opportunity for you given by Unacademy. Avail this properly and definitely you will achieve your goal. Okay, about myself, this is Anand Babu from Andhra Pradesh, Vijayawada. I worked in many reputed institutions. I have 50 years experience in teaching botany. I am qualified with MS BEd. I did my MSc in botany mainstream. My referral code in Unacademy, Babu Live. Okay, on my referral code, you will be getting what 10% discount. So don't forget to take a subscription. It is very, very important to you to have interaction with you in all aspects as far as subject concerned, covering all okay, question papers, previous and also model papers and also frequent tests, okay, as to analyze your skills here, to estimate your skills and to give perfection in all aspects. Okay, you will be having great opportunity here by taking this plus subscription. Okay, there is here plan for you, 1 year and 6 months plan. So choose any one of these. So already you can have experience in watching YouTube live classes, how they will be. You can estimate with all okay, our experience. We are ready to give right education to you. Okay, and here in this platform you are going to have many benefits here. There are regular okay, there are regular quizzes, okay, for self analysis, and also on each and every topic there will be a clear discussion and all of your doubts will be clarified. And one more I am adding here, completion on entire topic, on which discussion and what type of bits you are going to receive in that, and what are very important, okay, gray areas where you feel very difficulty. So all this will be explained in detail. Where there is a need, we'll give a conceptual idea to you. So avail it opportunity. And my advice is to take subscription. You will be okay, having great here benefits here. to achieve your goal successfully. Okay, shall we start with our program, regular program? And also one more point you have to remember, we have been discussing objectives only so far. Hereafter, I will take some of the very important topics on which there will be discussion. So be ready for that. Conceptual understanding is very, very important. Okay. Okay, are you following? Okay. We'll start with the first question in the anatomy of the flowering plant or plant anatomy here. Here is a interfascicular cambium here. Interfascicular cambium. Okay. Interfascicular cambium. What is this? Is it primary meristem, secondary meristem, or abnormal meristem, or none of these? So, one point here, the right answer is secondary meristem. B is the right option. So, meristems, you know very well, those are tissues. Plant tissues are divided into, classified into meristems. Am I correct or not? And also, what are they? Permanent tissues. Under permanent tissues, simple tissue, complex tissues. Okay, like that. Under simple tissues, parenchyma, collenchyma, mesenchyma. Under complex tissues, xylem and phloem. So, secondary meristem. Meristems are divided into, here is a primary meristems and secondary meristem. This is primary meristem. This is secondary meristem here. So, the primary meristems are formed early in the development of the plant. Primary meristems. But secondary meristems are formed so afterwards, later in the life cycle of the plant. Okay. So, under the secondary meristems, here interfascicular cambium and also cork cambium. These two are very important for secondary growth. You see here, plant is here. This plant, this is the initial stage. Afterwards, what happens if you take mango plant like that, it will, diameter of the stem will increase like this. Am I correct or not? So, for increasing the diameter or girth of this plant here, interfascicular cambium is very important. So, that interfascicular cambium comes under secondary meristems. Okay. These are formed later in the life cycle of the plant. That point you have to remember. Interfascicular cambium. We will study in detail about the secondary growth in plants. Then we will discuss about the interfascicular cambium. Okay. The right option is secondary meristem. Primary meristems are not. First formed meristems here. And these are nothing but apical meristem and another one. Apical meristem, intercalary meristem. Secondary meristems are, what are they? These are also known as lateral meristems also. Lateral also or cylindrical also. So, listen carefully. Regarding one bit, you may have several answers. So, you have to learn the concept first. Okay, well, let us go to second bit here. At maturity, sieve tubes do not possess. Sieve tubes here. What is sieve tube here? Sieve tubes do not possess. What is sieve tubes? The sieve tubes and what are the companion cells come under this phloem. These are the elements come under phloem. What is the right answer here? Sieve tubes do not possess. B is the right answer. So here this is the phloem. What are the components of phloem here? Main components here, sieve tubes. Okay. And companion cells. Companion cells. Okay. Those companion cells and sieve tubes are side by side present here like this. But sieve tube contains cytoplasm and also what is it? Vacuoles and cytoplasm, except what is that? Nucleus. Nucleus is not present here. I mean, initially nucleus is present in the plant sieve tube, and afterwards at particular stage the nucleus will be dissolved. Okay. So at maturity, sieve tubes do not contain water, these nuclei. But here companion cells are there. These companion cells contain this nucleus. But the function of these sieve tubes maintained by the, pressure gradient is maintained by the companion cells. Why? Because component cells will be having the nucleus here, and it is controlling the functioning of the sieve tube. So sieve tube is very important here. Am I correct or not? Made up of sieve cells, and food material will be okay. Nutrients will be transferred from one cell to another cell. That is another phenomenon. Okay, let us discuss. Okay, afterwards we will discuss that one. Okay, cytoplasm, vacuoles are present, cell wall is also present, but nucleus is absent. Okay, what is the next? Companion cells are. So similar type of bit here you see. Companion cells are parenchymatous in nature. So here I will tell you directly answer. This is parenchymatous in nature. Parenchyma, collenchyma, sclerenchyma. With which type of tissue, simple tissue, companion cells are made. Companion cell is very, very important now. Already I told you. And addition to this sieve tube here, companion cells are present. Those companion cells are nothing but parenchymatous cells. Okay. So it is a simple only. It is made up of parenchyma. Okay. Next. This is the fourth question. You see, presence of xylem vessels is a characteristic feature of. I mean, in which phylum you'll observe the vessels. Vessels, conductive elements. Am I correct or not? These are very efficient than tracheids. Tracheids also important for conduction of water and minerals. But vessels are most important, more efficient. These vessels mainly found in where? Developed plants, angiosperms. Angiosperms. So, presence of vessel is important character feature of the angiosperms. Here angiosperms, this is the right answer. Okay. In thallophytes, I mean algae and fungi, pteridophytes and gymnosperms, vessels are absent. Okay, but exceptions are there here. For example, if you take pteridophytes here, if you observe pteridophyte in Selaginella, vessels are present. Selaginella is a pteridophyte in which you will find the vessels here. Gymnosperms also in some of the plants vessels are present. These are exceptions. Exceptions. For example, Ephedra neatum. Okay, Ephedra neatum. So these are the plants belonging to gymnosperms where you will find vessels also. But usually vessels are absent in thallophytes, pteridophytes, gymnosperms. So these vessels are especially present in angiosperms. Vessels are efficient to advanced. Okay. These vessels only present in angiosperms. In all other groups, the vessels are absent, excepting some of the examples belonging to pteridophyta and gymnosperms. Okay. Are you following? Next, coming to this. Highly thickened wall and very narrow lumen. Very narrow lumen. Here the lumen is very narrow and highly thickened here. Highly thickened wall. Highly thickened wall here. So here I will tell you the right answer here. A is the right answer. So what are sclerites here? You know very well, simple tissues are parenchyma, collenchyma, sclerenchyma. So the sclerites belong to sclerenchyma only. Sclerenchyma. Sclerenchyma. In sclerenchyma, mainly two types of cells are there. They are fibers, okay, and sclerites. Sclerites, okay. Sclerites. So the sclerites are like this. Okay, the sclerites have thick walls here. That point you have to understand. Very thick walls. Why? Because these are lignified. Lignin is very hard material with which cell wall is made here, sclerate. Because of the deposition of the material here, the inner space becomes very, very narrow. This is what is called lumen. Okay, lumen is very narrow. Lumen narrow. Okay, sclerate is the right answer. Fibers also made up of that cell. What is that? Lignin. But here lumen is not okay, not very much reduced. And collenchyma, only in the corners deposition of cell wall material will be observed. In parenchyma, you see thin wall cells, cellulose only. Okay, so highly thickened wall and very narrow lumen are characteristics features of these sclerites. Okay, we'll switch over to another bit here. Sixth, component of phloem which is absent in most of the monocots. Most of the monocots it is absent. Of course, in monocots, in some monocots present. In most of the monocots here, what is the component is absent here. component of phloem. Phloem is nothing but made up of elements, seed tubes, and component cells, phloem fibers, and also what is that? Phloem parenchyma. Phloem parenchyma. Here you see phloem parenchyma is the right option. Phloem parenchyma is absent in most of the monocots. Especially it is present in where? Dicots. In monocots, especially, in monocots, phloem parenchyma is mostly not seen. Okay, phloem fibers are common. These are the parts of phloem here, component cells also, seed tubes also. Okay, except this phloem parenchyma. Are you following, ma? Attend regularly to the class, so you will be clarified everything. Okay. Next, coming to another bit 7. Anatomically, dicot leaf differs from monocot leaf in having. Anatomically, dicot leaf differs from monocot leaf in having. So, following 4 options are there. The 4 options here are there, which is the right answer here. Oh, in the beginning direct it is, I will tell you. Okay, more number of stomata on, okay, aboxyl surface than the addoxyl surface. Okay, that's, so this is a monocot leaf, and a dicot leaf, monocot leaf. Here you see in dicot leaf here, 2 membranes are there, no, inner membrane, outer membrane. Here inner membrane, aboxyl side, more number of stomata are present. Okay, than upper side, upper epidermis here. But whereas in monocot leaf here, monocot leaf are both the surface, upper layer and lower layer, that is upper epidermis and lower epidermis, stomata are present. Okay, that is the difference. So in this aspect, dicot leaf is different from monocot leaf. So more number of stomata on aboxyl surface means what? Inner epidermis. Then the addoxyl means outer epidermis here. So these are all common. Collateral and clothed vascular bundles. Okay. Dicot leaf. Okay. So these are right options. Next, coming to another bit here. The vascular chemium and chemium are examples of. Just now I told you, no, vascular chemium and chemium. That is, what is it? Examples of. Here, lateral meristems. Directly I am saying the answer, lateral meristems. Already I told you primary meristems and secondary meristems here. Secondary meristems. Under secondary meristems only, these are nothing but what? Secondary meristems, lateral meristems. These are also known as what? Cylindrical meristems. Cylindrical meristems here. So lateral meristems are very important. For what purpose? Where they are present? These are present in the lateral sides like this type of tissue. Parallel to the main axis. So these lateral meristems are very important. So here for the development of what? Vascular chemium and chemium. Okay, vascular chemium and chemium. So that what I mean, secondary growth of the plant will be taken place. This results increasing in the diameter of the stem. Diameter of the stem increases by vascular chemium. Okay, that point you have to remember. Chemium is very important in another aspect. Formation of cork like that. Okay, we'll discuss about that in secondary growth of the plant here. So apical meristems are not. They are present at the apex of the stem. Intercalary meristems in between permanent tissues. Am I correct on that? Elements of xylem and phloem. See here. Okay, this is also not correct option. Okay, coming to another bit here. The quiescent center in root meristem serves as. Here you see, I will tell you the right option here, that is C only. But you know very well, stem and root are there now. Both here are made up of here you see apical meristems are there here. Apical meristem. Okay. Here, see, this is meristem. Okay. But one more important point here you have to understand. Some of the meristematic cells, they lose their division. Just in the middle of the root, these cells are there. They lose the ability of division. They become permanent. Whenever there is need, for example, here, these are damaged. Meristematic cells which are present here around these are damaged. So in the damaged cells, in the place of damaged cells, new cells arise from these cells only. Those cells are present at the center. They do not divide, they become permanent. When there is a need, they again divide and form new cells, new meristematic cells. This happens only when meristematic cells are damaged. So, reserve for replenishment of damaged cells. Okay, important bit. This is reserve for growth hormones. This is incorrect. And site for storage of food which is utilized during maturation. This is also wrong. Region for absorption of water, wrong. So, no way other options are related to this 9th bit. So, right option is C. Next, coming to another bit here. Select the correct pair among the following. What is the correct pair here? You have to observe here. Right answer, I will tell you here. Dark color, no. Autumn wood, yes. This is. C is the right option. Here you see spring wood generally light in color. Okay. Light in color. But density is less here. Density is not more. Density is less. So this is not the correct option. And spring wood dark in color. It is wrong. It is what? Light in color. Just like this, light in color. Okay. This is light in color. And what is that? This is also wrong option. Okay. And here autumn wood is there. No, autumn wood. Autumn wood light color and high density. This is also incorrect. This is also incorrect. Sorry, this is also incorrect. Autumn wood, that is D is correct option. Autumn wood is dark color and low density. Okay. Autumn wood here, dark color, low density. So this is the right option. Okay. That point you have to. So one point you have to remember. In spring season, secondary xylem is formed. It is having more number of vessels. Vessel number is more. Okay, in spring wood. In autumn wood, vessel number is less. Why? Because in autumn there is no availability of water. In spring season only water is available. So vessels what are formed here, secondary xylem vessels, are very, number is more than here. Okay. Another important question here. Companion cells are 3 bits only from companion cells, you see. Companion cells are very important. Companion cells are associated with, so already this is explained, no? So here companion cells associated with sieve tubes here. But one point you have to remember, this is sieve tube, this is companion cell. Companion cells and sieve tubes are in the same origin. From single cell only they develop. Okay, that point you have to understand. Same origin. Origin is same here. That is also one more important bit under this companion cell. And also companion cell consists of nucleus. Another important point here. Important function that is concerned with the companion cell is to maintain the functioning of the sieve tube. Sieve tube will function properly in presence of this companion cell, without which sieve tube cannot function. I mean, for example, sieve tube is removed or companion cell removed. So both cannot survive. Okay, that point. So answer is what? Companion cells are associated with sieve tubes. Sieve tubes do not contain nucleus. This is wrong. This is wrong option. Okay, this is also wrong option. Again, one more bit here. Vascular tissues in flowering plants develop from. Vascular tissues in flowering plants develop from. Tell me now here. The direct answer I am telling here. This is C is the right option. Pleurum. What is that? Pleurum. What is pleurum? Here at the apex of the stem here, or root here. According to histogen theory, outerly one layer will be there, that is dermatogen, And here in between, here just below it, what is it? Another peribulum is there, peribulum. And finally, innermost layer, this is what is called pleuron. Pleuron. So this pleuron is responsible for the development of what? Vascular tissues. Vascular tissues develop from pleuro, which is the innermost layer of the apex of the stem. According to histogen theory, middle layer here in between this dermatogen and what is it? And pleuron. Another one is a peribulum is present. Dermatogen is important for what purpose? Important for giving epidermis. Epidermis of the root to stem or leaf like this. And another one is peribulum. This gives cortex. Okay, according to histogen theory, this gives what? Vascular tissue. Our correct option is pleuron only. This is the answer relevant to this 12th question here. Okay. And cholenchyma cells are absent in the stem and the leaves of many monocotyledonous plants. It is so because cholenchyma cells are absent in stem and the stem and the leaves of monocots. Leaves of many monocotyledons. Okay now. What is the reason here? Why cholenchyma cells are absent in the stem and the leaves of many monocotyledons? The right answer here is one point I have to understand. Sclerenchyma, this is the D option is right. Sclerenchyma appear at an early stage of the development. In monocots, sclerenchyma presence is very, very important. Monocots are very weak, stems are weak. Easily plant will be affected by external factors like this. So sclerenchyma presence is very much required here. Why? Because the sclerenchyma thick-walled, made up of thick-walled cells. Okay, no? This is thick-walled cells here. Parenchyma. So it gives mechanical strength mainly. So mechanical strength to monocot plant will be will be offered by the sclerenchyma. So sclerenchyma appear at early stage of the development in monocots. So cholenchyma cells are absent. Cholenchyma also important for giving mechanical strength, but not as much as a sclerenchyma. Okay, that point. Cholenchyma not only giving mechanical strength, other important functions are there, giving flexibility to the plant and also tensile strength. Flexibility. Okay, flexibility. When high blow of wind, attacked, so the plant parts bend. They do not. They bend, do not break. That is another question. Don't worry about that. And which of the following composes the hypodermis of the dicot stem? Here, what is the meaning here? Which type of tissue is present in the hypodermis of a dicot stem? Parenchyma, cholenchyma, sclerenchyma, and aerenchyma. Here, dicot stem, no. Easily remember cholenchyma. If it is a monocot stem, that is a sclerenchyma. Parenchyma, no. Parenchyma cortex is made up of parenchyma. This is not right answer. And the sclerenchyma is found in where? Monocots only. Monocot stem. Just below here you see. This is dicot stem. Just below the dicot stem, what is it? Hypodermis is present. This hypodermis is nothing but cholenchyma. Okay, cholenchyma is not spelling here CH. This is not correct. CH, cholenchyma. Cholenchyma here. Whereas in case of monocots here, just below the epidermis here, what is that? Sclerenchymatous cells. Already I told you in monocots, sclerenchyma well developed. Sclerenchyma. This is sclerenchyma. Okay. In case of monocots it is. Okay. In case of dicots it is. Okay. Cholenchyma. This is dicots. Dicot stem. Right. Monocot. Okay. So aerenchyma is also. This is not correct. Why? Because aerenchyma is found in hydrophytes only. Okay. So this is the right answer. Next, coming to a vascular bundle consisting phloem and cambium on both sides. Both sides. Sorry, here the spelling mistake. sides of xylem is called. What is that? Here I will directly tell the answer, that is B is the right option. Bicollateral vascular bundle. What is bicollateral vascular bundle? Here this is the vascular bundle. Okay. This vascular bundle. Here what is that? Phloem and cambium on both sides of xylem. Okay. Vascular bundle consisting phloem and cambium on both sides. This is xylem. Xylem is surrounded by what is it? Phloem. Okay, this is also phloem. Xylem is surrounded by phloem and separated by cambium here like this. This is cambium. Okay, this is outer cambium, this is inner cambium. Here this is xylem, this is phloem. So vascular bundle consisting phloem and cambium at both sides of xylem. This is the xylem. Okay, on either side of the xylem, what is there? Phloem is present. Outerly cambium also present. Outer cambium, this is inner cambium. This type of vascular bundle is called bicollateral vascular bundles, which are generally seen in cucurbita species. Cucurbita. Okay, like this. So the radial, okay, it's not correct here. Okay, bicollateral 2, it's not right. And amphi vessel, it is also not correct. So collateral also not correct. Here right answer is here the bicollateral. Okay. Next, coming to what is it? The vascular bundles present in the what is it? Midrib of a dicot leaf. This is right. This is a direct question here. In midrib, what is the size of the vascular bundle? Here you see, right option is direct answer. This is largest. In other parts of the leaf here, vascular bundles are smaller when compared to the when compared to the vascular bundle that is present in the midrib region. So these are all wrong options here, and this is only the right option. And next, coming to main function of lenticels is. What is the main function of lenticels is? Lenticel. What is the meaning of lenticel? Important function of the lenticel is here. I will tell you exchange of gases. Here one point you have to remember. Plant after secondary growth, what happens? It is completely occupied by water. Bark formation will be there. So completely it is sealed. Outer surface is sealed. But how oxygen will be available to it? Respiration, for respiration purpose, oxygen how it is available. But here and there, here you see parenchyma cells inside form lens-shaped openings. They are called lenticels. Small pores through which exchange of gases takes place. Okay. So main function of this lenticel is exchange of gases. Transpiration also occurs through lenticels, but transpiration rate is very little. That is 0.0 like that only transpiration. So it is not the correct option. Okay. This also guttation. Guttation is nothing but different process. It is not passing through stem. It is passing through leaves, not by stomata, through special openings. So bleeding, it is also incorrect answer. And another question here is Collenchyma differs from sclerenchyma in. What is the difference here? So right option here, possessing protoplasm even at maturity. But here is what is the main difference between collenchyma? Collenchyma is a living tissue. Protoplasm even present at maturity. But whereas in sclerenchyma, all cells are dead. So it is a dead tissue. Okay. Sclerenchyma is completely meant for main function of it is giving mechanical strength support to the plant. Okay. A monocot stem with secondary growth is. Here generally secondary growth is not seen in monocot stems. But in one of the following options, secondary growth is seen. This is this is what is called yucca. Okay. So another important bit here, see 20th bit. In which of the following plants sunken stomata are formed? So we'll complete here 20 bits today. That's why I exceeded some time here. So have little patience. So thank you very much for your cooperation here. You are still okay intact with the subject. Here, in which of the following plants sunken stomata here? You see, sunken stomata, nerium. Direct answer is nerium. Nerium is a xerophyte. Here you see stomata are not exposed to deep inside present like this. This is due to control the rate of transpiration. If stomata appear on the surface externally, rate of transpiration will be more. Rate of transpiration will be reduced here because of the arrangement of the stomata. The stomata are sunken stomata, deeply present inside, sunken. So this is an example which is a xerophyte where we will find sunken stomata. Thank you very much. Okay, thank you very much. I am going to end up and again with a new topic. Some of the concepts we'll discuss next onwards. Okay, tomorrow also there will be discussion only on what is it, MCQs on a particular topic important for NEET and other competitive exams. And also I will tell you what are the important concepts which are very much important. Okay, thank you very much.